let's continue with our last session then. The next talk will be given again by two people, and there's Shaoxia Chen and Christos Savva. The reason for now being two is that because both of them run our EM facility, and it's, I think, a good idea you all know them by face. So Shaoxia will start.
Okay, thank you, Shaox. I think today I will talk to you about a little bit practical thing. Past one to nearly 2 weeks, you're releasing all the lectures, and you got a lot of information from the theory to how you do the processing, and then you got a lot of example from the application. So today I was going to talk a little bit about what we have in our facility, and later Christos was talking about the training we usually provide. So this will be the outline. I get these few slides, the following slides I'm going to talk about. So, and yeah, this is some kind of really happen, in everyday life here. So I think it's good to people who have an overview of what we have and how you choose the right instrument, how to book the facility, how to access it, and how to get the training. So with all this kind of policy and knowledge, I think you can make your dream in LMB become true. So what we have here, it's quite simple. We have a 5 microscope, TEM microscope for data collection, and then we have a series of other small equipment device for prepare sample and do all sorts of things, get ready to the microscope and after microscope. And also we still have some digitalization machine for the film. So this is 5 microscope we have. I think maybe a lot of them you can recognize which one is which, but when I put up a file, I could not really recognize these three. So we have this bottom one, basically it's our medium resolution microscope. Yeah, you get. So from here we got 2 split. So this is no cryo, general use, this cryo, and we got a 1012 cryo and no cryo there. And then when I take picture, nice it there. So I take one just nice. And no, this one is T12. And then this is Polara. Also, and when we come cryo, so we don't know what happened inside. So now I put up this table just to summarize what different of this 5 microscope. So in here, I have 2 groups of the microscope. This is the medium resolution microscope. The sequence like from the previous slides, so from here, and then until the end. So in here, I put in this sequence. So this is name and the room. On here, in this slide, so for this medium resolution microscope, you can see the energy, electron energy is not very high. The range we can from 60 to 120 keV. The three are same. So for this one is a higher one. It goes up to 300 keV. You got more energy. So the next difference is for the first one. The first microscope we use for general use, that means we use for most of the negative stain work and the sectioning, the room temperature sectioning. So the next 2 machine is capable of do cryo-EM as well because they have a cryo-EM holder for them. And this middle one is the 10-9 split cryo-EM. We can do more high tilt. You can have a high tilt single tilt holder as well. So we can do more work. So basically, these 3 microscope we use quite more. It's very intensive for check all the sample and optimize the sample preparation. So then we come to the high end one. Is our data collection machine. So we have a high voltage, high energy, and then we have the electron source are few emission gun. It's compared to the 2N one. This is the tungsten one. So then this few emission gun give a very high bright electron being and then very high coherence. From Chris' instrumentation talk, you will see this. And the next one is the difference between these two machines is that the cryo get much more higher parallel illumination because it got more length for the condenser. But basically, if we say these two data collection microscopes are quite similar, we both have an energy filter for doing tomography, for reduce the inelastic scattering and get a better contrast for your sick sample. And then we also have we all have Falcon 2 at the pre-give position, so do the single particle correction. This Falcon 2 is a direct electron detectors, so we can hear other people's talk, so we can reduce the noise quite a lot. We use the new detector. Then we all now the DQE. We already now it already much better than the film we usually use. So now we not use the EM film anymore. So, but they are still okay for using EM film. But from last year we not really use the EM film. So there's another different, except that this have an illumination system different. We can easier to achieve the parallel illumination. But on parallel you also can achieve. Although it's not easy, but you can do a careful setting. You can. Then for another different is the post-GIF is very different. After the GIF on the parallel, we have a Cannone ultra scan 4000 CCD. It's a 2K by 2K CCD. It has to be very good, a unique combination in the cryo-EM world. But now we have another better combination. We have a Cannone K2 submit at the post-GIF position at the cryos. So this one is another direct electron detector. And then the others are some small camera for help. And then we have different software come with all this machine, operate this machine. So, but the basic difference is, well, they're all same. You can all have different control machine, and then you can have two different tomography acquisition software for these two data correct machine. We also have the tomography software and in this cryo spirit. So theoretically, you can run the whole series and check your tomography sample and in this machine. And now we introduce more software like TechNice scripting. Now we can install more like series EM in most machines. So it will be very good machine. You can do your training practice and check your session and your tomography data at the first view. So yeah, when we have this 5 microscope here, so on my next slide is going to talk how to choose the right machine. I think from the combination of this the instrument, we can all see say when you need to optimize your sample, check your sample. Of course, you start from this end one, because they're easy to operate, and then you just need to pop in and don't need a long session. You already know what your sample look like. And for this whole the two data collection machine, it's quite a long queue as well, and then it takes a long time until you find your sample to set up and until you find your sample is good or not. So it's more efficient so you select the right machine at the beginning. I put down here is like when you do the screen for negative 10 and room temperature, of course, you go to switch general. This is a very easy machine, and well, I don't know, the CCD is breaking down. I hope come back soon. But this machine, people just jump in and out. Although we say booking, there's some you don't book for too long, but people maybe just go to half hours and book in and out. So they're very good machine for use. And then if you need to check cryo, these two machines and then are available. And Source has just showed us a picture from Julia. He can look at a very small particle, 200 kD, even 200 kD in cryo. You can see clearly in the spirit cryo, the CCD. So it's not just say you have to go to Prara to check your sample for small particle. With your good sample preparation, you don't have too much in your buffer, like and a good concentration. You're still able to see from this machine and save you a lot of time. So and then we soon have this tomography ready, so people can use this one more before you move to data collection machine, because they are very long queuing. This one perhaps 3 weeks, 2 or 3 weeks, but this one at least 4 weeks. So because I count about 2 weeks ago, we have 29 active user, internal user. So it's not like you put a name, group leader put a name, that they really come to use. so that means if everything okay, you get 1 session at the month, and then but if microscope break down or have some software problem, you really limit your time there. So this is our three machine. So but if you don't not sure which machine to use first, you're welcome to talk to us, and all our group leader, they all have quite good idea which I want to start. So, and the next one is the booking policy. Well, this is not very popular, but we have to book something up. Well, first, of course, you really have to change before we go to use. We don't want to make anything like a human error to delay our time, so we are all available for here. And then I think the new user, of course, you should talk to us, yes. But otherwise, we don't know what really carry on, because sometimes when the machine have problem or when we do engineer need to do something, you maybe we have to change something, so we have to let us know, and then we can this our contact. That with all we have all in this information available in the microscope room. Then we have some booking rules, some guidance for booking. So this one is you only book 1 session in advance. That means you book a microscope 1 session, but you have to finish this session before you book the other one. Yeah, book the other one. Of course, you go to that day, maybe you start the book already, but don't book 2 sessions in 1 microscope before, then give people more chance to get in to book it. Well, sometimes you can book at the data collection microscope, then you can book other microscope, it's okay. But as soon as you are not overlap, you really use it, and then you can. But this is for each microscope. And then how long the session is, we defined for the general use, and for next then for the three negative dam microscope, I think half day is okay. 4 hours is quite long for people taking do a good check for a lot of grade. But usually people don't need that long. 2 hours, 1 hour, then even jump in half hours in between. So they all work. But for the Cryo-EM, we know the setup and pumping time, you need to take time. So when you change your grid, it takes 1 hour in this one. So we say you can book for a whole day. Yeah, whole day. So the whole day will define different how late you can stay. So this, and then get ready for cryo cycle for next morning is okay. But for the 2 data collection microscope, each session we say it's defined 24 hours. So it's like normally 9 o'clock to next morning 9 o'clock. So it depends on how you're going to use. Usually you start loading your grid at the morning and set up until the lunchtime. Then you will get already rough idea what grid you have. If you have a good grid today, and then you set up EPU or do your manually low dose collection. And at the night time, if we can use EPU do the automatic collection for the single particle or your tomography batch. For the batch program from FEI or your series EM also can do now. You can set up something for overnight, or maybe some people like just stay there, watch it, then stay overnight. We also often see the student people just sit there, have a shift every 4 hours. So this is the cryos, and the weekend bookings is now. Now it's a big. It has been okay until recently. We found some confusing. At the beginning, not everyone can do the weekend booking, especially. But for general microscope, they're okay. People are capable of use. But for the Pollara and cryos, you really need to be dependent to use before you can book the weekend. And also, that time only one or two or three people can use, so it won't count. But now I think more people can use Capo over weekend, and then we all count the EM session in the weekend as one session now. So I hope people are following this. But sometimes if you really lost one session, lost several sessions together, so maybe one of the weekend we can squeeze out, let you have. But we really just we can work on volunteers because this is what the situation we have. So okay, this is the booking. And then what people can help. Me and Crystal here. We are here to happy to help you, or if any information you want to get, include the inquiry training. And then we have a very good research group here, and a lot of people support all different parts aspect of the Cryo-EM facility. Like Greg, they have a lot of the detector, developed a lot of the detector and a lot of computer data handling. He's very good at this. So he knows every network how you do it. So you can always get a very good advice. And we got general advice how to use microscopy image processing from all our research group. We can Richard here from a long time. Then he know everything. And then we got Tony, Nigel, and Shaoxia and Lori for all different image processing. I think when you are new, come to a new EM project, especially talk to one of them to get the idea what you can get from there, what you should start. That's a really good idea. And then if any other problem when during the microscope, when you're at the microscope you need help, or when you're at the weekend you break down, is anything you can get. We have the contact telephone number in the EM log. You can get Christos, and then you'll notice that like Venus, Chris, Termite, they're all around all the time. Well, not all the time, but sometimes when they're in the lab, you really can get help there. Then they have very different expertise. Like if you want to do, Venus most a lot of single particle stuff. Where Chris, you can cover a lot of things if you don't need graphing and other carbon coating stuff. He has a very lot of experience. And Tom might do a lot of tomography and at the K2, so it's very useful source. And Jill Howard, she did a lot of things from cell biology to long temperature sectioning and others as well. So she's got a good contact point from the cell biology people. So other sources say, well, we get some electronic one, the EM one. We have something, but haven't really put up. We have written something, but there's some very good information everywhere already from the LMB wiki and from every EM group's website. So if, for example, if you go to wiki in the internal website, you can see they already have quite a lot of lab. You even get 3D EM wiki, a lot of information, and Schultz has one, Rory has one, other people in here. They all have different kind of information. When click in and find quite a lot of very interesting things. Like you go to, this is a source group one. You can have how to do the rely on how to do cryo-EM. There are a lot of information here, and then you got like Lori, you can even have tutorial in EM. So you will need it from the start, any other information. So this is what I have, I think. So the next one I will pass to Christos. He will talk about the training.
So I'm sure everybody here is excited to get their hands on the microscopes now and get a structure. So I'm just going to give an overview of the training on the transmission electron microscopes, what you will learn, how long it will take, just so you have a realistic expectations of what needs to happen so you can start using the microscopes. So we generally break up the training into three stages. The first stage, you learn how to use the microscope and align it properly, take good images at room temperature. Once that training is complete, you'll learn the basic cryo-EM sample preparation and screening techniques. And once you have that down, or you're feeling confident, we'll move on to the high resolution data acquisition. So the first part, the use and alignment, takes place on one of the 320 kV instruments. It generally takes about 10 hours total, which is usually spread between 3 and 4 sessions. Sometimes you'll finish a bit earlier, sometimes it takes a bit longer. And we prefer if people can at least have one session a week. Sometimes we can accommodate more depending on the microscope availability, but at least if you have a session every week, it's less likely you forget something and the whole training will finish faster. So for training on the small microscopes and general use, just contact me. During this time, you're going to learn the different TEM parts, the lenses, apertures, CCD, an overview of the vacuum system, and Chris Russo gave a very nice overview of how the electron microscope works. It would be good to go back and see that video of the lecture as a kind of theoretical revision of before you go into the practice. You will also learn how to load a side entry specimen holder for room temperature, how to do the alignments on the microscope, and how to take nice images. So nice images meaning your sample might not be great, but you'll have at least nice on rings and the alignments will be performed correctly. So if required, and if you need to, I can also show you how to use a carbon coater to evaporate thin films of carbon, how to then prepare negative stain grids, glow discharge, and so on. So once you've got the general basic TM usage down, we can then move into the cryo part. So this can take a little bit longer. It requires at least 4 sessions and probably additional time for the ancillary equipment. And at this stage, one can actually start screening their own sample. So we're going to be doing all this cryo freezing, plunging, loading cryo holders onto the microscope. So we might as well put a specimen that's worth looking at, and at this point you can start looking at your own sample. So to get trained for basic cryo-TM, you contact me again. This also takes place on the 120 kV instruments. So during this time you're going to learn how to prepare cryo-EM specimens, and you'll learn how to use the FEI VitroBot to plunge freeze your specimen. You'll learn how to use a side entry cryo specimen holder and how to prepare it. It needs a bit of preparation, a little bit more preparation than the room temperature procedures. And you'll also learn how to use the LODOS software, which is part of the FEI software, which we use to collect images of being radiation sensitive specimens. And this was outlined in Laurie's lecture. So once you've got this down and you're more or less independent and you've got a nice optimized sample, then we can go on to the 300 kV instruments and start collecting some data sets. So this will require multiple sessions to become fully independent. And like Shaoxia mentioned, that we have a lot of users now. I think we have about 70 total users in the LMB and over 30 which use the 300 kV instruments. So there is a little bit of waiting time. I think the current time is about 3 weeks for the Polara and over 4 weeks for the Cryos. So the sessions will probably be spread apart, but you will combine the training sessions with data collection. So for training on these microscopes, you can contact Shaoxia or me. So during this time, you will learn how to load grids into cartridges, and then how to load these cartridges into multi-specimen holders which go into this microscope. So advantage of the Polara and the Cryos is that you can load multiple specimens at once rather than just one at a time for the 120 kV instruments. And you'll also learn how to align a field emission gun microscope. Luckily, the software interface with all the FEI microscopes is very similar. There's a few differences for the Polara and the Cryos that accommodates the loading of the specimens, but a lot of it is very similar and the basic principles are the same. And you will also learn how to use this APU software, which is used for automated data collection. It's been present on the Cryo since installation, and we recently installed it on the Polara as well. So we can do automated data collection on both microscopes now. So at the end of this course, I guess you're going to ask yourself, is cryo-EM suitable for me and my protein of interest? So I think you've learned a lot during this course, and there's 2 main considerations. One is, and this, like Laurie mentioned, is the sample suitable. So it has to be relatively large, although smaller complexes are becoming possible nowadays. And good biochemistry is very important. So you don't want to take something to the microscope that you're not very certain of. You want to get the best purity, homogeneity possible before you start the microscopy. The other consideration you have to take into account is, do you have enough time to do this project? So you can see the timeline here. It can take up to 5 months to start becoming independent on the 300 kV microscopes. After a couple of months of the basic training, you might be able to start collecting data here if your sample is ready. But overall, it's not really a very fast process. This is the best case scenario, really. And I think Rebecca is going to talk to you about how things go if everything goes pretty smoothly. But it's just something to consider. This shouldn't be really treated as a side project. It needs a little bit of time commitment. But Shaoxia and me are happy to help you to get started and take you through the process. So, thank you.
Thank you, Shaoxia. Thank you, Christos. I think if there's questions about how to use the microscopes and so on, it's best we just come and chat to Christos and Shaoxia. So unless there are no burning questions, I suggest we move on to Rebecca. And as Christos just mentioned, sometimes there's best case scenarios and they sometimes do come true. And I think Rebecca's case is a close approximation of that. So.
Right. Okay. So I'm going to tell you a little bit about my experience of solving a structure using electron microscopy, and this is from the perspective of someone who's really brand new to the field and was trained just as Christos just described, and as Shore said, everything went really, really smoothly, so we've ended up with this structure relatively quickly. But the structure I'm going to talk about is a structure of a mammalian ribosome bound to the translocon, and in mammals, the most important part of the translocon is called Sec61. And what you can see here actually is one of the early electron micrographs of this complex, and this was taken in about 1966, but similar ones were done a little bit earlier, and they're actually used to identify ribosomes. So you can see here these little granular kind of spherical particles. They're attached to the ER membrane via the interaction of the ribosome with this Sec61 channel. And this is a really important complex for understanding how membrane proteins are inserted into the plasma membrane and how secreted proteins are translocated across it. And so what I'm going to tell you about today is how you can take a sample that was actually made really similarly to this one from 1955 to get a high-resolution EM density map and then eventually a near atomic model. And I think as a brand new user, I need to preface this by saying that none of this would be possible, particularly for someone like me, without the tremendous facilities we have here and all of the people. This is just really a small portion of the people who have really put a lot of time and effort into making sure these facilities are accessible and for training people like me. So special thanks to Christos for helping me get started and really showing me how to operate a microscope. So what I'm essentially going to go through is just touch on each step in the process of going from obtaining a sample to having a final structure. And so I think probably the most important part of any structural biology project is picking the right sample. And this can mean different things for different techniques, crystallography or EM or NMR. And we took a sort of old-fashioned approach to this structure. So we actually purified the ribosome Sec61 complex from a native source. So in this case, it was a pig pancreas. And the reason we did this is because there was evidence that when you tried to assemble the complex in vitro or from recombinant components, that actually the channel was falling off the ribosome and or was flexible. And so we reasoned that if we could actually minimize the number of manipulations and the time that it took to go from literally the tissue in an animal to being frozen on an EM grid, we might have a better chance of getting high resolution for the complex. And the problems with using native sample are many, but I think kind of the main ones are that they're just not as clean as something that you can make recombinantly. There's inherent heterogeneity in the sample. and you have this added burden of trying to identify the components in your final EM map. But we figured that there are strategies to overcome all of these problems. And at the end of the day, having a sample that was very, very stable would be more important than trying to deal with these issues downstream. So with a sample in hand, I had my first microscope session with Christos in early January. And as Christos explained, the first several sessions on the microscope are just learning how to operate it. And I like to refer to this as the how not to break the microscope phase of training, which is very important because people get very angry when you break the microscopes. And at that point, we spend some time actually learning to make cryo-EM samples. And I think one of the things I didn't appreciate before starting this project is how many different components actually go into making a sample for cryo-EM. So you've heard several times from Lori and Christos. So you have your grid, maybe you want to put carbon on it, which has to be made. You have to glow discharge the grid, you have to add your sample, you have to freeze the grid. And all of this actually takes a fair amount of time to kind of get your head around and to do in a moderately competent manner. But Christos is very, very patient. And so in kind of late January, I was able to actually put some of my samples frozen on a microscope. And I think we probably had about 4 sessions in the microscope where I was simultaneously learning how to operate a microscope for cryo-EM and also optimize the grid and sample preparation. And I think this is maybe one of the most important parts of any microscope project, because I think if you do a good job here, it'll save you a lot of time downstream. And for my particular case, this was important to determine basically all sorts of things about both the sample and the grid preparation. So what detergents to use, how much detergent, the buffer, trying to optimize the ice thickness. And all of this kind of operated in parallel. So we wanted to pick conditions that were best for both the sample and for visualization by EM. And I think in mid-February, or early February, I actually had some nice images of my sample by cryo-EM. And so you can see very clearly the ribosomes on the grid. They're relatively well dispersed. There's some clumping problems, which we think were as a result of the detergent and having a membrane protein attached to the ribosome. But it seemed like it was nice enough to actually try and collect a data set. So I had my first session in the Polara in late February and was able to collect a very small data set, or moderate-sized data set, I guess, of about 280 images, resulting in about 18,000 particles. And at this point, I turned to Israel, who helped. I had other funnier pictures, but I thought they were not appropriate. Who helped do some of the data processing that you've heard about today, because it's certainly one thing to hear about it in theory, and then it's a whole other thing to actually try and implement it in practice. And having someone who knows what they're doing is a big time saver. So we ended up with a very low resolution reconstruction of these 18,000 particles. So this is about to 10 angstrom resolution, and this map hasn't been sharpened, and no 3D classification has been done. But the exciting thing for us at the time, and it's still kind of exciting now, is that you can see this really clear donut hanging off of the large ribosomal subunit. And of course, what that is the detergent lipid micelle surrounding the Sec61 channel. And we weren't going to learn a whole lot about the biology for something like this, but what we did know is that the factor that we were interested in studying was there at relatively high occupancy. And so this seemed like a reasonable sample to try to collect a high-resolution data set. So for that, I turned to the cryos in early March. And the sample was suitable for using this automated data image collection software that you've heard from Shaoxia and Christos. And this is much, much faster than me. So in the same time on the Polara, where I could collect about 300 images, I ended up with 1,500 images and about 80,000 particles. And just to give you some idea of kind of the scale of all of this, that I think I certainly wasn't really expecting. This is a huge amount of data, so about 1.7 terabytes. And this is just only kind of a mid-sized data set. Some people are working with things that are much larger than this. And not only is it a huge amount of data to store and manipulate, but it also takes a huge amount of computational power to actually do all of those image processing things that you've heard about over the past 2 weeks. So these are just all of the different steps in image processing, and in total, takes about 9.2 CPU years to deal with 80,000 particles. So it's actually not that many particles, so it's a tremendous amount of computational power. And it really wouldn't be possible without the tremendous cluster we have here at the LMB. So Jake and Toby owe a big thanks. And I'm not going to go through each one of these steps, but I just wanted to point out these two, because these were particularly important for my project, because we knew we were using a native sample, so there was going to be some heterogeneity there. So the first 3D reconstruction from this large data set used all 80,000 particles, and you ended up with a map that looked something like this, that went to about 3.4 angstrom resolution after sharpening. And the really exciting thing about this is that not only could we see what looked like high-resolution features in the ribosome, but also we could really clearly see the transmembrane helices in this Sec61 complex hanging off of it. And if you can see carefully, not only can you see kind of sausages, but it actually looks like the sausages might have some features. So it seemed like maybe we were going to actually be able to get something biologically interesting out of a sample like this. So that was a very exciting time. And then because we knew we had heterogeneity, you did a 3D classification and essentially ended up with about 4 classes of different biological states. And the most interesting of which went to about 4 or 3.5 angstrom resolution. And just to show you a local resolution map that you heard about from Richard yesterday of kind of the highest resolution classes. This is of the 60S ribosome, a subunit bound to Sec61, and this is the 40S subunit. And what you can see is that the core is very well ordered, so to about 3 angstrom resolution or better, and regions of Sec61 are at similar resolution. And then the resolution drops off as you get a move away from the ribosome in Sec61, and you can see kind of residual detergent or lipid bound to the channel in pink here. And the 40S subunit was to somewhat lower resolution. But all of this processing took about 2.5 weeks, and at the end of March, we had our final EM maps that we could use for model building. And the density was good enough that it allowed Israel and myself to place the ribosomal RNA, side chains of the ribosomal proteins, and the Sec61 channel. And actually what I think is really cool is to place about 160 magnesium ions in the structure. So this is probably the absolute best resolution map I've ever seen of a ribosome by crystallography or otherwise, although I haven't seen Alan's maps carefully, so his might be slightly better. But essentially we did all of the model building as Alan has described. So they were built in COOT and refined using RefMac. And this resulted in a complete near-atomic model of the ADS ribosome bound to this Sec61 channel. And so if we go back to my little timeline here, so I had my first microscope session in January, and as of yesterday, we have finally finished building the structures, and they have been deposited in the PDB. So this has all gone very quickly, although in large part this model building portion at the end went very fast because of kind of a heroic effort by Israel to really build a really nice ribosome structure, because the ribosome is really, really big. And with that, I just have one slide to tell you, so why do we think this is an exciting structure? What have we learned? So as I said, we have a near-atomic model of the mammalian ribosome. So this is the highest resolution mammalian ribosome that's been solved to date. And we think we've done a really nice job of producing a nice model for this. So hopefully this will be useful for future structural studies and biochemical experiments and that sort of thing. In part because we used a native sample, we actually had a population of ribosomes that were actively translating. So we could see a nascent peptide in the ribosome bound to a tRNA and entering the Sec61 channel. And so that is definitely one advantage of using this native sample from an active tissue. And the resolution was sufficient to really allow us to analyze the detailed interactions of the Sec61 channel with the ribosome. So we can kind of explain exactly how those ribosomes get attached to the ER membrane in that image from 60 years ago, where they first saw the ribosomes on the ER. And so with that, I think hopefully I've shown you that EM can be a very powerful technique. Sometimes it can go very quickly, and hopefully it can allow you to study interesting complexes that would be intractable by most other methods. So I will just end by thanking everyone again who was on that first slide at the beginning of the talk, and just want to say thank you to Manu, who is my boss and allowed me to kind of go off and try and learn something totally new, and also to Christos and Israel for working really hard to try and get these images and produce these structures. Shores for a tremendous amount of help with the data processing. Every time I had an error, I was in his office asking a stupid question. And Venki for his support and encouragement throughout the entire process. And you for your attention.
Thank you, Rebecca, for your exciting talk. Perhaps we have time for one or two questions, if there are any.
So why is your detergent not uniform around the translucent?
Say it again.
Why is the detergent belt not uniform around the translucent? I saw only a small...
Why is it only towards the end? I basically just lowered the threshold to get rid of most of it. But if you or increase the threshold, if you lower it, you'll see the entire micelle, as well as some of the other components of the translocon that we know are there because it's a native sample. But it's easier to look at it this way.
Paul?
In what way was the Sec61 in your structure different from your prior model?
In what way was it different? Well, so there's no mammalian crystal structure of a Sec61 channel existing. So that was new. And what really wasn't known was how the channel interacts with the ribosome, because there had only been low resolution models built to about, I think about 9 angstroms was the highest. So those were new and were built de novo in the maps.
So you didn't have a non-mammalian model of Sec61?
No, yes. So there is an archaeal crystal structure and a bacterial crystal structure, but there are no mammalian crystal structures. So they're homologous in kind of general architecture, but actually side chain density and sequence is somewhat different. And in particular, those loops that interact directly with the ribosome had never been seen before beyond 10 angstroms. So.
Okay. I think in the interest of time, we'll move on to Vina.
Okay, as the last speaker, I've been asked to explain what happens really, some realities on when you try to do EM. So it's not always things work straightforward. So for the past 2 weeks, you have been listening to the theory of EM, and you would have realized that if you had a large molecule, then it's easy to work and it's easy to get a high resolution structure faster. So it largely depends on how accurately you can orient the molecule. So if you have a very low orientational accuracy, then you get high resolution structure with less number of particles. So here I have a scale of difficulty which starts from 0 to 10. I don't call anything 0 because you still have to make a grade. If you don't have a good grade, you won't get good images. So it goes like this. The smaller particles are more difficult. And so I put some of the examples we know. For example, if you had a virus or any other highly symmetrical molecule, say up to 4 megadalton, then they are relatively easy and you can get structure very fast. Ribosomes come next. And then you have intermediate structures about 1 megadalton, which you can do faster. Beta-gal is a kind of standard test specimen we use, which is about 440 kilodalton. Although I put at 6 here, simply because not everybody can get high resolution with this sample, which you can get from Sigma. So a lot of people are working on that. Only 2 labs I know can reach high resolution with this. Then you have TRPV1. So BSA, monomeric BSA is 67 kilodalton. You can see it on the micrograph, but you can't still orient. So I put it at scale 10. So this is. So I thought I will explain. I will take 2 examples of the protein complexes I work. Both are membrane proteins. Both cannot be crystallized yet. So complex 1 and the acetylcholine receptor, which is smaller than here. So complex one, as most of you know, has two domains, which is a matrix arm soluble protein, and then the membrane arm, which is hydrophobic. So the bovine complex one, which I work, has 44 subunits, and it's about 1 megadalton. So this is a typical example of a micrograph of bovine complex 1 in detergent micelle taken with the cryos. So I didn't screen on the Techna, I directly went to the cryos simply because I thought my grades will be good, and so it was. So and once you are more experienced, I think you will be more confident on putting your grade directly on the cryos. So this, so you can see that there are different views of complex 1 in ICE, and this is in collaboration with Jaipeng and Judy in MBU. So you see the 2D class average with different views, and these were used to generate an initial model with EM2, and then you get a map about 5 angstrom, which shows 3 different things. The blue is the detergent lipid micelle around the transmembrane region, the brown is the iron sulfur cluster, and then you have the protein molecule in gray. So if I take the detergent lipid densities, then you can see a lot of transmembrane helices crisscrossing the membrane domain. And it's more important to validate, so I've done all the validation methods recommended by Richard, tilt pair analysis, high resolution noise substitution, and then comparing the final model to the map also. And so in the local resolution, although the overall structure is about 5 angstrom, you can see that the core of the protein is close to 4.5 and so on. So this couple of examples of densities showing some of the transmembrane region have helices with side chains visible, and the beta strands are starting to separate. In this case, you can see beta seed here and that alpha helix from a soluble protein. So this is the overall model. We are able to build about 70% of the protein molecule into this map, even at 5 angstrom resolution. And this allows us to assign quite a few supernumerary subunits. The core subunits are here in gray, and then the supernumerary subunits, which we are very confident are highlighted. And there are certain transmembrane helices, which are single transmembrane helices, which we still can't confidently assign, so we have left this for now. So now people generally ask me 2 questions. Why is it at 5 angstrom, and how do you improve it? So this graph, you can read the theory behind it in Peters and Richards' publication in 2003, which shows that for a given B factor, what kind of resolution you get. So this is pyruvate dehydrogenase, which Peter and Richard worked on. These images were taken on film, and then with it has a icosaedral symmetry. So you can see that it had a B factor of about 1000, and it gave about 8.7 angstrom. So these are 4 different examples which I show here, all taken on direct electron detectors. Then you can see that complex 1 is about here, around 5 angstrom. Then you have these 3 other examples. So I put the number of particles here for each of the specimen, and then the asymmetric unit, because hepatitis B virus has a icosaedral symmetry and beta gal has a D2. So then you can see the number of asymmetric units used in refinement, and then kind of nominal resolution. So you can see that the complex 1 with this number of particles is comparable to others, and I do have a better data set, about 40,000 particles to 4.6. So it looks like nothing is wrong with it. So the other thing to remember is all 3 data sets here are taken in similar microscopic settings, so that it's easy to compare. So then, if I had the same B factor and same conditions, and I don't change anything, how much number of particles do I need to get to say 3.5? So it looks like if I use this B factor to calculate, then you can see that for every 0.2 angstrom from 5, if I want to increase, then the number of particles doubles each time. So this is the worst case scenario, because the B factor will improve as the map improves. And so this is what I think is kind of a guide, but it's not a rule that it's going to follow this here. And this is very useful when you're trying to collect a lot of data, and it's better to pause and think what is happening, and if I add more particles, is it improving or what is happening? Or if your PI comes and asks, where is my structure, then you can say, I'm here, and then I need to take this many number of particles to get it to here. So for example, now I decide I want to go to 4.2 from 5, which is like 300,000 particles. So I calculated I need roughly 10 days of data collection with the cryos, which is like 30,000 particles per day, and then 15 days of computing with 250 processors. This means that with a communal instrument like we have here at LMB, this is roughly about a year of data collection, because I get one session per month, which is actually not true. I get more. So complex one is a kind of a very straightforward project, because it worked with only 2 days of data collection in the cryos, and then few days of processing, and then modeling took longer than that. But there are examples which I saw. So this is an example of membrane protein again, which Nigel has been working for a long time. He has used this helical crystal to get a model of acetylcholine receptor, and it's very abundant. It's heteropentamer, and it doesn't have a symmetry. It's about 250 kilodalton. So I've been trying to look at this single particle and then see if we can get better than 4 angstrom. So this gel showing a very nice band, blue native gel showing the protein, the receptor in detergent, and then this is a Falcon image of acetylcholine receptor. When compared to complex one, you can see how small it is. Then if I show you the reference-free class average, then you can see that it looks more like the receptor. And then in the side view, you can see the detergent belt is very strong, and then it's more like a featureless blob without any structure attached to it. So one interesting thing is that this thing which Nigel modeled, the alpha helix on the cytoplasmic domain, is lost when you treat with detergent. So which means that you have only this part here, and which I think is one of the reasons why I have an accuracy of about 8 degrees and the resolution is stuck around 16. So if we plot acetylcholine receptor in this graph here, then you can see that, so I've used only about 8,000 receptor molecules. It's about 16 angstrom, and a B factor maybe around 2,000. So how does one improve it? So there are 3 different things I thought are important. So one of the problem I have is that the receptor likes to stay in one orientation. And I found that using different detergent does manipulate that. So it's possible that finding the right detergent will give better distribution. Then of course, change the shape and features of the molecule using, say, for example, antibodies is one possibility, so that it's bigger. And also you have some feature to orient. And then the last one is detectors in counting mode, either K2 or Falcon. But these will not be helpful unless I'm able to somehow find the best out of this. So even the best detector will not give you any higher resolution if you don't have the right sample. So to summarize, so if you had a very big molecule, and say, for example, with highly symmetrical, then it's possible to get a structure within. So we have tried this with Hep B. You can get a structure within 20 hours from the time you put your grid to the last refinement. And it's possible, so for example, with complex one, you can do it in days now to say 5 angstrom. And for the smaller molecules, it takes generally from months to years. And there is this gray region where you still can't orient, so it's going to take a few more years for the techniques to develop. So Cryo-EM has a lot of potentials, and we hope that most of you will happy to use it. Thank you.
Thank you, Vinod. It will be a great comfort to others who are struggling to show that you show something that doesn't work. So, are there any questions for Vinod? Leo?
So with the smaller molecules, is there, like you said, wide theoretical limit to the precision of the orientation determination, or can it be theoretically improved?
Yeah, I think there is no limit. I think Richard can tell you that you can orient as small as 40 or 50 when the techniques develop. But I think with the present advancement, I think 150 is the limit, I would say.